Teaching Chair at the Semel Institute for Neuroscience and Behavior and Department of Psychiatry at UCLA. His studies utilize pharmacological and non-pharmacological perturbations, functional neuroimaging, electroencephalography, and computational modeling to map body perceptions associated with natural and experimentally induced stress. Central aims of this work are to discover modifiable neuroscience-based treatment targets for anxiety, eating, and stress-related disorders, to develop clinical tests to identify these targets in individual patients, and to design neuroscience-based therapies capable of ameliorating the symptoms and signs of mental and physical illness. The floor is yours, sir.
Thank you for the invitation to be here. Just to start out with some disclosures. So actually, the title of this talk was sort of purposely intended to stimulate your thinking. So I'm not necessarily going to delineate what the next generation biointerfaces are. In fact, I think you're already doing that here at this institute. But I would be interested to hear your thoughts as at the end of the talk in relation to how these kinds of interfaces might be relevant for interoceptive neuroscience. And I promise you, by the end of this talk, you'll be much more, you'll be interoception experts, or at least feel like you know a lot better what that concept is about. Can you hear me okay? Is it too loud, or is it just fine? Okay. So I have no disclosures associated with this talk. I will say that at the middle of 2024, I moved from the Laureate Institute for Brain Research back to UCLA, where I did my training. And that institute actually has some parallels to the Terasaki Institute. And a lot of the work that I'm going to show you actually came from the work that I did there. Of course, none of this happens in a vacuum, and I just want to start by thanking the various lab members, alumni, collaborators across multiple institutions and funding sources. But again, a special thanks, especially to Xiling for the invitation. We met each other in the fall of last year, and it was great to kind of connect and to be here as a result. But especially also to Mehmet, Yangshi, and Alicia. It's been a good set of discussions so far. So I want to start. These are sort of the summary and take-home points. If you remember nothing else, you know. First and foremost, and this is in the abstract that was described for the talk, that I want you to come away with the idea that interoception provides a direct channel into hidden physiology that is relevant for shaping emotion, cognition, and health. You'll see pretty clearly that the tools to access this domain are still somewhat rudimentary, but that by using controlled perturbations of interoceptive signals, that would be, in my opinion, an optimal way to sort of look at how these internal sensations are encoded across the brain and body. And then that will have relevance for design principles for next generation biointerfaces. So that can do things that are relevant to the mission both of this institute, but also the broader mission that I have in my lab for psychiatry in terms of enhancing well-being, personalizing treatment, and really just understanding how brain-body connection leads to cognition. So we're going to zoom out a little bit, and I want to start with pointing out that we have a growing problem. We have a brain parts list that is increasingly sophisticated. We can now catalog genes, cells, and map circuits with extraordinary precision. Here's the example of the mouse brain atlas. But we also have a growing body parts list. We have efforts for a human cell atlas. We have the entire full body transcription factor for C. elegans. Now, having these delineations, having all of this information, doesn't actually explain how a living system maintains itself and how it feels from the inside to optimize recovery. And so how these parts come together to regulate homeostasis and generate feeling states, particularly in the setting of disease, is what I call the integration problem. And it's definitely the next frontier in science and medicine. So my argument, my central thesis is that to understand that, we need to study interoception, which is this bridge between conscious experience and body regulation. And I promise, again, you'll know a lot about what that is by the end of the talk. Interoception is not a new concept. It was actually defined back in 1906 by Charles Sherrington, who got the Nobel Prize for discovering or conceptualizing the synapse. But what you can see is that the field of interoceptive neuroscience, like that of cognitive and affective neuroscience, has sort of emerged and, let's see, I don't know how can you see my mouse? Really, it's only been in the last sort of 20 years now that there's been kind of an explosion of interest in understanding how the nervous system represents and subsequently regulates signals from within. And a couple things that I would like to point out, not the least of which was the shared Nobel Prize by Julius and Patapoutian for discovering some of these sort of so-called interoceptors that are receptors for internal bodily stimuli. So clearly there's a lot of interest in this area, but as you're going to see, this is an area that is in its infancy in many ways. This is not my baby. I pulled this from the internet. But as you're listening to this information, you'll probably come up with some questions and realize that there are some gaps and opportunities. And that signifies that there's incredible potential for growth, but also, much like a baby, many ways in which progress could be prematurely stunted. So just a visual reminder of the importance of keeping this early developmental stage in mind. Now, to introduce you to interoception, I understand, how many of you have actually heard that term before? Excellent. So, fresh slate. So, to start this off, I want to introduce you to interoception through, I want to take you back 10 years about to 2016. So, with the support of LIBOR, I convened the first international meeting focused on interoception and understanding its role in mental health. The aim of that meeting was to unite a multidisciplinary group of scientists to try to accelerate progress in understanding and conceptualizing research on interoception with a focus on improving mental health outcomes. So we came out with a special issue and a white paper. These are all the various people who participated in it. And we developed essentially the first consensus definition of interoception. And that is that it is the process by which the nervous system senses, interprets, and integrates signals originating from within the body, providing a moment-by-moment mapping of the body's internal landscape across conscious and unconscious levels. So we'll let that kind of roll around in your mind a little bit. It's a process, so it's dynamic. Nervous system, sensing, interpreting, integrating signals from within the body for a moment-to-moment mapping. And then we have the woolly mammoth across conscious and unconscious levels. If you look at research on interoception over the past 100 or so years, what you'll find eventually is, particularly in human samples, another commonly studied term, interoceptive awareness, which is the conscious sensation, interpretation, and integration of information about the state of inner body systems. So inner body, internal landscape, what are these systems? What are these signals? Well, quite a bit. Cardiovascular, respiratory, GI, bladder, hunger, thirst. You have a whole host of blood markers that are being measured here at this institute. You can divide these into several different categories. So they vary both with respect to, this actually says non-painful over here, and then you have painful over here. A way that I would illustrate that is, anybody ever have a kidney stone? Or how much pain was that? 0 to 10. 10, right? 10, 11, 12. Worst pain in your life that you've ever experienced. Well, this is an electron micrograph and an X-ray image of a kidney stone. Imagine this guy is the surface of it, shredding its way through your ureters, right? You don't know what your ureters feel like, but when you've got something like that going through, it's an unambiguously salient stimuli, right? 10 out of 10, you will know, you'll not be able to focus on anything else, right? So one of the features about interoception is, if you think of it as an iceberg, and you know that most of an iceberg is below the surface. So most of interoception is actually unconscious, and the part that is conscious, interoceptive awareness, is really just the tip of the iceberg. Okay? So most of what we think of as interoception, you don't actually consciously feel, even if it's guiding your behavior, even if it's guiding your function, even if it's facilitating your maintenance of homeostasis. One of the things that we added to the literature was said, okay, well, interoceptive awareness in and of itself isn't a monolith. You can divide it into different components. It's multidimensional or multifaceted, right? If I ask you to feel your heartbeat right now, you would first have to pay attention to it and then note whether or not you feel it, right? If you feel it, you might ask yourself, is my heart pounding? Maybe you recently had RSV, or you're about to give an anxiety-provoking talk. This is not one of them, by the way, but you all know what it feels like for your heartbeat intensity to be elevated. Where do you feel it in your body? How well do you actually feel it? We'll come back to that. Interoceptive accuracy is actually one of the most commonly studied facets of interoceptive awareness. Finally, this concept of insight. When you are pretty confident in feeling your interoceptive signals, how much does that actually map to the state of the body? Sometimes it does, and sometimes it doesn't. And there are various other elements that are worth that can be delineated, including self-report scales, where you just give somebody a systematic set of questionnaires to assess. So if you look at the literature, you'll find it littered with studies that focus sometimes unilaterally on one or more, but not all of these dimensions. And that's one of the arguments, is that it's important to do that if you're interested in this prospect. Another piece is that our internal organs are not really merely physiologic workhorses, but they act as broadcasters, sending sophisticated signals that are mostly unconscious. But as I said, when something happens in the body, when homeostasis is disrupted, they come into sharp focus when the body's physiology is disrupted or under threat. So palpitations, dyspnea, vasomotor flushing, these could be associated with a variety of different affective or physiologic states that can be differentiated from other ones in, say, the gastrointestinal domain. Okay. The other piece of this is that these signals have a valence and arousal component. So they can not only be painful or non-painful, they can be pleasant or unpleasant. So to make this case clear, a few years ago I had a chance, I was visiting a colleague in Utah, and we landed in a blizzard, and he took me snowshoeing at 8,000 feet, 3 feet of snow in a couple hours. Very happy side. Heart pounding, had a great time, right? Positively valenced. However, the opposite is true for patients in our anxiety disorders clinic at UCLA, where pathological avoidance of cardiac sensation and arousal is pathognomonic with anxiety. So our patients routinely, for the same degree of physiological arousal, say exercise, something that is physically good for you, can cause intense anxiety to the point where they avoid any type of situation or activity that they think will put them in that state. Okay? So the other piece that's important from a neurobiological perspective when you start looking at interoception is that it's part of a brain-body feedback loop. So what you have is organ systems speaking to nervous systems, but then the nervous systems, they speak back. And you have multiple systems, right? Cardiovascular, respiratory, GI, et cetera. So interoception can be thought of as this sort of monolithic component. You've got genitourinary, chemoosmo in there, and then immune system. We haven't really delineated, but within the nervous system, of course, there's central, peripheral, autonomic, somatic, and enteric components. And then, of course, there's the microbiome, which is a whole another aspect that we won't talk about, kind of a world within a world that also plays a role in manipulating your interoception. When we look at the neural circuits of interoception, we're not talking about networks in the brain. We're talking about circuits that span internal organ systems from sensors through pathways to networks focused specifically on function. So you can think of a neural circuit for interoception related to a function, whether it's appetitive, affective, arousal related. And finally, with the awareness component, These are related to distinct networks that are not about identifying objects such as plants or living beings or other people. They're always self-related. When you experience interoceptive awareness, it's your own body that you're feeling. It's not anybody else's body, right? Which is very different than if all of us were to look at, say, a red apple in front of us and to report on the signal that we perceive. Interoception is fundamentally self-oriented. Now, we know a lot about peripheral receptors and pathways that are relevant for the transmission of interoceptive signals to the brain. This is a review paper that kind of provides a nice job of outlining a schematic of these. And one thing that I would emphasize is that you have sort of multiple pathways. You have a vagal pathway, you have spinal pathways, you also have cranial nerve pathways that are connected to distinct cell types, to distinct stimulus-specific receptors, mechanoreceptors, chemoreceptors, et cetera. So when we talk about interoception, we can lump all these things together in a very general way, but then it becomes important to sort of specify for a particular disease or a particular function, what's the channel that you're looking at. Of course, in the organism, all of these signals are being represented in real time, and there isn't a single brain region involved in interoception either. So this is from a review we conducted a few years ago looking at the literature, and it turns out that it's not simply the insular cortex, which is listed schematically here, but there's actually a variety of other areas, and some of the data that I'm going to show you will speak to these different brain regions across different domains or axes of interoception. And what this says is cerebellum ? And others ? So likely this is an incomplete map that will be updated even further over time. For example, the cerebellar vermis likely plays an important role. The other piece that I want to emphasize by mentioning this paper from 2 years ago now is that there's a very specific and rapid way that the brain has access to certain interoceptive signals. This study made a lot of headlines because it showed that there are mitral cells in the olfactory bulb of mice that actually show spiking activity within a 20 millisecond period of the pulsatility, which means that the brain has access to signals about the heartbeat 3 times faster than it gets from the peripheral cardiovascular system in terms of your aortic and your carotid body receptors, okay? So why would your brain need to know what's happening at such a fast time scale? If there's time, I can sort of give you an evolutionary argument, but this is probably one example of multiple forms of interoceptive signals that the brain probably prioritizes evolutionarily privileged access to very rapid and quick information about what's happening inside the body. There's also an interesting problem related to multisensory integration of these signals. So this was a paper that showed that humans without a sense of smell actually show different respiratory patterns. So obviously smell and breathing are interrelated, but the mechanics of breathing in these individuals was also different. So there's quite a bit of rapid, immediate, and complex individual and multisensory integration happening. Now, I'm a psychiatrist, and most of what I deal with are not biomarkers in the clinic. In fact, in psychiatry, we don't have any real biomarkers that we can regularly rely on the way people in cardiology or gastroenterology or various other fields, cancer biology, can utilize. We diagnose conditions based on symptoms. Usually when a disorder is a collection of symptoms. Usually when you identify the mechanism, it gets identified as or categorized as a disease, say Alzheimer's disease, Parkinson's disease, and then sometimes it moves out and goes to neurology or other specialties. But symptoms are what we deal with, and you can think of symptoms as having a type of hierarchy across conscious and unconscious processing, where you can have both conscious symptoms as well as unconscious symptoms. And the question and the fundamental goal that I have in my research is to identify where do interoceptive symptoms come from that form a bulk of many psychiatric disorders, and I'll illustrate those for you in the next slide. Do they come from the body, so this sort of unconscious loop? Do they come from the brain? putatively this sort of conscious network, or is it the interaction between the two? And I would advance that there are probably instances where it's one or the other or both. Okay? But back to symptoms in specific psychiatric disorders. This is the first textbook on interoception that was published a year and a half ago. I wrote the mental health chapter, and what I outlined here were symptoms that are relevant here in this middle column. These are all diagnostic symptoms that are utilized to diagnose these various types of disorders. Depression, eating disorders, panic disorder, GAD, PTSD, substance use, borderline, autism, etc. The ones in blue are the ones that I've mainly studied in my lab, but the list is actually growing. I don't have schizophrenia in here, but there's actually a good amount of data even beginning to show abnormalities of cardiac interoception in schizophrenia. So to get at this question of differentiating brain from periphery or their interaction, I would argue that perturbing or manipulating peripheral organ system signals is an optimal way to understand how interoceptive neurocircuitry causes symptom perception to occur in a disorder-relevant fashion. So if it's anxiety disorders related to fear of heartbeat sensations, can we manipulate the heartbeat? If it's abnormal gut perception in eating disorders and fear of gut sensations, how can we perturb gut signals? Okay? So I'm going to show you work related to each of those domains. The first one is going to be related to heart-brain communication. And I want to start by asking you a question. How many of you are confident that you can feel your heartbeat right now? Raise your hand. You? Okay. And if you were to err on the side of caution, not caution, but just sort of be optimistic, how many, best guess, you think you could probably feel it? A little bit more, okay. So probably about a third of you indicated at some degree that you think you could feel it under resting conditions like this. Well, if I put you to the test in the lab, you'd be pretty correct, right? But only 1 out of 3 can accurately sense their heartbeat signal under resting conditions in which people have been typically studied in psychology settings. Think about it from a visual neuroscience perspective. If you're trying to understand visual processing or the neuroscience of reading, if only 1/3 of your people can read the letters, how are you going to identify the neural circuit for reading, right? So there's a fundamental problem, and this doesn't matter whether you're an advanced meditator, they're no better, that was the first paper that I ever published as a grad student, or if you have somebody else's heart beating in your chest, there's still sort of this fundamental floor effect, right? But we all know what our heartbeats feel like, right? And I gave some of these various examples: exercise, anxiety. If you go further north and you go to Six Flags and you love riding a roller coaster, or during sexual activity, right? Like, we all have a sense of our heartbeat sensations during a perturbation. So the question that I had is, if we have this fundamental floor effect, can we use a perturbation to actually give everybody that conscious sensation unambiguously and then measure brain activity? So to do that, I adopted what I call a visceral psychophysical approach to studying interoception, leveraging knowledge of the hypothalamic pituitary adrenal axis. And of course, we're not talking about measuring cortisol at this point with a contact lens, but what I focused on, which was very interesting, but what I focused on is the other path of the output from the adrenal gland, looking at epinephrine and norepinephrine signaling to the brain. And in particular, taking advantage of a molecule called isoproterenol, which is a close analog of epinephrine. Difference is that it doesn't cross the blood-brain barrier. It very focally stimulates beta-1, beta-2 adrenergic receptors to perturb cardiac and respiratory signals in a very particular fashion. We give it intravenously, and what you can see is that when you do that, this is an average curve in 21 healthy individuals under double-blinded, placebo-controlled conditions. You can see that you get a nice dose-response curve. The normal heart rate in a human is from 60 to 100 beats a minute. And you can see that at a 0.5 microgram dose or a 2 microgram dose, you can get a nice boost in the heartbeat signal that you don't see with the saline. And then we can see that it has different phases: a peak phase, a recovery phase, an anticipatory phase, etc. So that's just manipulating the signal. How about looking at perception? So what we do is we pair that with a dial, and we just have people rate in real time the intensity of the sensation. You can see that on the right axis here, and that's the purple line. Again, this is under blinded conditions. This is just showing the 2 microgram dose. You can see that when you have this perturbation, you get a very nice change in interoceptive awareness that tracks, even with a little bit of a lag, but still tracks the feeling. And then again, we can divide this into different phases. Peak and recovery relative to a baseline. We can do really nice dose response curves for the physiology, and then dose response curves for the perception here on the top right. And what I would point out is that a 2 microgram dose gives a very noticeable perceptual change. A 0.5 microgram dose, even though you can see that it gives a nice heart rate change, less perceptible, but perhaps still present. And we'll come back to that in a little bit. When we look at detection curves, whether people binary say yes or no, they feel something, now I can take everybody in this room and I can make everybody feel their heartbeat, even when you're sitting in your seat. And then we can study that. We can look at neural processing. We can localize where people feel it in their body. Now, if you were to try to predict what area of the brain is relevant for this, this is the kind of map that you would come up with. A multi-level hierarchy, starting down in the heart itself, going through intrinsic cardiac ganglia. This is an 8-level hierarchy that we came up with that processes this signal in different ways, sort of chops it up and divvies it. And you can see that the insular cortex is here. You could sort of superimpose an automatic, non-conscious, and conscious layering. You have here areas of the cerebral cortex involved in executive cognitive control, default mode processing, et cetera. And so the question is, what area of the brain, when you make this perturbation, shows the strongest response? So we adapted this approach to the fMRI setting. Here's an example of a participant in the scanner with a nurse infusing the drug. We're watching them and their signals in the scan room, and they have this sort of speedometer dial where they're rating the sensation. The first study that we did was actually 10 years ago at UCLA. We found that, again, that you could do this nice perturbation. And across the brain, what we saw was dose-related activation in the insular cortex over here, and you can see that listed here. And we used actually 2 sequences to sort of confirm that, an arterial spin labeling as well as a bold signal. Okay? So that gave us some novel evidence that the insular cortex is at least an important cortical way station for this conscious processing. But remember I mentioned that there's both vagal and spinal pathways for interoception. And when I was developing this method, I would often get a lot of questions about, well, you know, where do you feel it? What's the receptor? What's the pathway? You know, sometimes I feel my heartbeat in my chest. Sometimes I feel it in my neck. Sometimes I feel it in my temples. You know, how do you know that the skin and spinal pathways associated with the skin mechanosensation aren't also playing a role? So we did a study to look at these pathways. We found a patient who had very unique, due to herpes simplex encephalitis, had acquired bilateral damage to the insular cortex. A few other brain regions as well, but for this purpose, he was missing his insular cortex essentially bilaterally on both sides. Very rare, but the hypothesis is that if the insular cortex is the only area that's involved, when that's gone, he shouldn't feel it. So he's got the visceral pathway missing. If he does feel it, maybe there's somatosensory afferents that are playing a role. So we did this study. The main thing that I want to point out to you is this is his name was Roger. This is Roger's heart rate. He shows a nice heart rate response similar to the healthy comparisons. Here's the healthy comparison perception. And you can see that his perception, even though it's delayed, is intact from an amplitude perspective. He felt this was actually a 4 microgram dose, not even the 2. He felt the 2 as well. I'm not showing that. But we had unambiguous evidence that even in the absence of that sector, he could feel it. We then said, okay, well, where do people localize the sensation? We used the topical skin anesthetic, lidocaine, to attenuate or knock out the mechanosensation. And now in Roger, we have both pathways missing. The healthy individuals still have an intact insula, so they should still be able to feel it through that route. And here's what we found. The healthy individuals could feel it, and Roger felt nothing. Okay? And we validated that. We were asking him questions in real time. His heart's pounding away at 125 beats a minute. He couldn't feel it. So, so this provided some causal evidence that you have multiple pathways for interoception. And if you think about it from an evolutionary standpoint, again, the brain would want to be privileged to having multiple routes for having access to such a fundamental signal. Now, we've done other studies looking at clinical populations. This is a study in generalized anxiety disorder that we published a few years ago, where we used the same method, but our main question was, do patients with generalized anxiety disorder have a hypersensitivity? You know, they report more aversion to feeling heartbeat sensations. Do they have a greater sensitivity? And it turns out at the low dose, this is the 0.5 microgram dose, you can see that they both reported greater awareness of the sensation, but they also had greater heartbeat change. So there was this element of both maybe greater adrenergic receptor sensitivity, because isobutyrenol only acts in the periphery, combined with a perturbation that then reaches the brain, perhaps augmenting their sensory awareness. Now, our hypothesis was that the insular cortex was going to show hyperactivity in the GAD patients. But to our surprise, even though it showed dose-related increases, no differences between the patients and the healthy comparisons. Okay? We did see a group difference in the brain, but in a different area. This is in the ventromedial prefrontal cortex. And again, this was only at that 0.5 microgram dose where we saw the physiological and interoceptive awareness differences. And the difference in activation mirrored closely the degree of activity. So we have evidence here that there's both sort of the insular cortex does play a role, but perhaps in the setting of affective pathology and anxious pathology in humans, maybe the medial prefrontal cortex showing more of a role. Now, this was quite interesting in relation to a paper that came out of Carl Deisseroth's group a couple years ago, where they essentially did what I like to call the mouse homologue using optogenetic methods. They created mice with carmine that basically could, when they injected carmine channel rhodopsin into the heart and they used the fluorescent light, they could actually drive the mouse heart rate, which is normally at around, I think, around 500 beats a minute, to 600, 800, even 1,000. And then they could use tissue clearing to see which areas of the brain were activated. Now, it turns out in the mouse, it wasn't simply optogenetically increasing heart rate that activated the neurons that you see here, posterior insular cortex and medial prefrontal cortex. It was actually in the setting of optogenetic pacing of the heart plus anxiety induction using various threats of shock, open plus maze, things like that. So, you know, if you look at these sort of studies side by side, the posterior insular cortex area is homologous to this dysgranular insular area that we activated in our study. And then in MPFC and VMPFC, again, homologs. What was interesting in this paper, when they inactivated the insular neurons, they could abolish the anxiety-like phenotype, whereas in this inactivating medial prefrontal cortex, that had no effect. So there's maybe some evidence that, you know, from an anxiety standpoint in humans, medial prefrontal cortex is maybe carrying more of the weight in terms of the anxiety-related contributions. A question that I have is how could targeting this region, the VMPFC with neuromodulation strategies, define this region's role in interoception? Could you use it to help patients better interpret arousal from the autonomic nervous system? Could it be a potential therapeutic? And there's multiple ways of doing it. Real-time fMRI neurofeedback is a non-invasive way. TMS is a non-invasive way. I know you had Jeff Deskalakis speak to you a few months ago. There is actually a way to do TMS to the VMPFC using temporal interfering fields like dual stimulation. I spoke to somebody. Another way is transcranial low-intensity focused ultrasound. And I'm just going to briefly highlight that approach here, because what it can do is, unlike TMS, it can get at deeper structures, deeper than the cortical surface, and also much more focally. Here's a paper, if you're interested in this from a disease standpoint, looking at sort of different ways of targeting. Actually, I'm going to skip that for a second. here's an example using focused ultrasound where basically we were able to stimulate the posterior insular cortex and modulate a cortical signal called the heartbeat evoked potential. It's an EEG signal that is considered to be a correlate of cardiac interoception. Another colleague of mine is taking a different approach where instead of targeting cortical regions, he's targeting subcortical white matter to try to stimulate a circuit or really a network of different cortical regions and showing some evidence of changing thalamic and VMPFC connectivity. So I want to be mindful of the time. I'm going to skip on to another set of studies that I think you'll find interesting, and this relates to perturbation of gut-brain signaling. So another area of work in my lab focuses on eating disorders, and one of the things that we know for sure is that individuals with anorexia nervosa have quite a bit of abnormal gut signaling and abnormal symptoms related to the body. So if you're not familiar with anorexia nervosa, it's a severe psychiatric condition that has a high mortality rate. It causes severe food restriction, is associated with substantial weight loss. Patients have intense fears of gaining weight, and they also have abnormal body image. So when they look at themselves in the mirror, they might have a very low body weight, but what they see in the mirror is actually a much larger individual. Now, these are the diagnostic symptom criteria for this disorder. The only real diagnostic biomarker in all of psychiatry right now is this weight loss. If you have low body weight, that's part of the criteria for anorexia nervosa. That's not a very sensitive one because you're just stepping on a scale, and somebody might be a marathon runner, or they might have cancer cachexia, right? So simply looking at body weight in the context of these symptoms is not sufficient. It's been well described that these patients have cardiometabolic and gastrointestinal dysfunction, bradycardia, constipation, electrolyte disturbances, but they also almost unilaterally report things like feeling full prematurely. So they eat, you sit down and we had a nice Subway sandwich. I felt very satiated by the end. After 3 or 4 bites, they might say, "Oh, I'm full." You're like, "What do you mean? You just had like 3 bites. How is that possible?" They might report abdominal bloating or cramping. And these are sort of common symptoms associated with the condition. So the question is, what's the best way to sort of access that? I was inspired by a study from 56 years ago. This was only in 3 patients. But what they tried to do was look at abnormal interoceptive perception. And what they had patients do is, sorry, let me just show you. What they had people do is swallow a tube. They anesthetized the back of the throat. They had them swallow a tube, and then they instilled 1 milliliter aliquots of a milkshake substance into the stomach, and they said, "Do you feel it? Do you feel it? Do you feel it?" Now, this was in 3 subjects. And they did see a difference in the perception, but it was not compelling by any means. And of course, how are you going to get this kind of approach in a psychiatric clinic, right? There's no way you're going to do it. There's also no way that you're going to be able to do more invasive things that have been typically studied, like having people swallow an electrical probe where you can deliver electrical stimulation, or inflate balloons, or place balloons from below in the rectum. That's never going to translate, right? So when I was asking this question, I came across an approach that was developed by a company called Vibrant, which is a vibrating capsule. So this is a company out of Israel, and they were interested in a different question, solving the chronic constipation problem. In their idea, this is a capsule, it's got a battery and a motor in it, you swallow it, and after 7 or hours or so, when it's roughly in the vicinity of the colon, it starts vibrating. And their observation from some preclinical work was that delivering this kind of stimuli increased gut peristalsis. So maybe the buzzing made the colon peristalsis happen, it squeezes more strongly, and people poop better. I asked them if they could change the stimulation so that it would start going off a few minutes after they swallowed it, and could we use it in a similar way for visceral psychophysics. So this is an example. We used a signal detection assay to look at normalized A prime, So we could see that when we delivered what's called a normal stimulation, this is the amount of stimulation they delivered for their constipation studies. People would feel it above chance for the most part. And then when we delivered enhanced stimulation, they felt it more accurately with a reduced response time and not a clear change in the electrogastrogram signal. We validated this with what we call the gastric evoked potential. So we can't do fMRI scanning with this, but we can use EEG. It's one of the reasons I'm interested in EEG assays, to see that, in fact, there was a potential in the brain that showed a differentiation between these two signals, the normal and the enhanced. And of course, you can see that overall, you get a very large stimulation across the 3-second duration during which this stimulation's happening. It's localized to a region of the brain in the posterior midline, sort of the parieto-occipital area. The amplitude of that stimulation correlates with perceptual accuracy or interoceptive awareness. This is from a different study, but that looked at the correlation between the gut rhythm via the EGG signal and brain activity in resting state fMRI that showed, in fact, you did have a little insula activity and somatosensory, but most of it happened in this posterior midline parietal occipital region, just beneath the surface of where we saw this potential. It's a different potential than a cortical evoked potential, which is much more rapid. So that you'll notice that this stimulation happens with 400 milliseconds after this differentiation between the two signals, whereas this one happens within the first 200 milliseconds. So different potential. When we were publishing this paper, we were asked to replicate the data. We replicated the finding. Then we were asked, well, you know, these capsules move around in the gut. How do you know where the capsule's located during the 30 minutes of stimulation? So we did some abdominal X-ray imaging, looking at where it was in the different segments of the stomach, as well as the small intestine. This is just an X-ray example of a capsule in somebody's stomach in the antrum, just about to exit into the duodenum. And what we showed is that most of the stimulation is occurring in the gastric and the duodenal segment of the gut. So we felt fairly confident that we had a relatively focal segment of the gut that we could stimulate with this mechanosensory input. We could change sensory signals. We could change symptoms. And now we wanted to do a study looking at anorexia nervosa. So we had an R01 to do that. And the paper is actually under revision right now. But this is just some of the top-level results where we took inpatients with anorexia nervosa. These were females. It's predominantly a female condition relative to healthy comparisons. And what we did was we said, okay, what's their perceptual awareness? And it turns out that when you deliver the normal intensity of stimulation, patients with anorexia nervosa are much less likely to feel it. They have lower perceptual accuracy. Their visceral acuity, if you will, they don't see the stimulations the same way healthy individuals do. But when you give a strong enough stimulation, within the same block, we do counterbalance blocks of normal and enhanced, you can rescue that deficit. Okay? They also tend to miss the signal more often. This is just, the miss rate is not a parameter that enters into this equation. So we have multiple instances where when the capsule is there, they're not sensing the stimulation that it's providing. We're also interested in moving beyond these sort of standard signal detection measures. We've used Bayesian computational models. Is anybody familiar with Bayesian modeling? Raise your hands if you are. Okay. So I won't go into it in detail. All I will say is that the value of Bayesian modeling is that it gives you the ability to evaluate not only the sort of classic sensory input sort of finding from sensory neuroscience where you shine a light and okay, the light has this peripheral transduction pathway, it activates a signal. We know that maybe that happens the first time you see a signal, but over time you learn about visual input, you learn about your sensory signals, you formulate predictions and prior sort of inferences about the conditions in which you would expect to see a signal. For example, if there were no, in fact, you kind of can't really see outside of this part of the room, part of the building. Right now, without looking outside a window, it could be midnight right now, pitch black outside. or it could be noon, broad daylight. And if you didn't know, if we were in here long enough that you lost track of that, if I told you it's noon, you would have an inference, oh yeah, it should be light outside, right? So computational modeling allows you to sort of look at that immersion or fusion of sensory input and predictions. We developed a paper to look at a series of modeling approaches to look at different components of that, interoceptive priors, learning rates, which are how your priors change over time, and precision being sort of the accuracy of the mapping of true vibrations to the belief you have about them. So if I told you, for example, it's actually 5 p.m. right now, you would form a different inference about, well, okay, well, maybe it's not fully bright out, maybe it's a little bit dimmer, right? Maybe it's around sunset, right? So interoceptive precision gives you that. Long story short, patients with anorexia nervosa had stronger initial priors against detecting the vibration signal. They had lower interoceptive precision. They had lower learning rates. So when the vibration happened, healthy individuals were more likely to form inferences about, oh, okay, there should be another one coming soon. Patients were less likely to do that. And they were more likely to assume that a vibration was not coming from periods when there was no vibration. So abnormalities there. I want to just point your eye to this graph on the right. This is kind of interesting. Patients report much less hunger before the task. Both groups showed increased hunger afterwards, but the patients actually had a larger increase in hunger rating. So simply stimulating their stomach led them to feel more hunger, or at least rate feeling more hunger over time. Still lower than the healthy comparison group. But imagine this is a group that's kind of disconnected with their appetitive sensation. Could we use this in a stimulatory fashion to train them to detect hunger signals more accurately to then improve eating behavior? Okay, almost to the end. I'm very interested in sort of predicting outcomes. How can we use these markers to get a better sense of how people are going to do in the future? Using sort of a cancer staging model, we developed a classification system, a standardized classification for relapse, remission, and then recovery. Sort of, are you in the red for relapse? Are you in remission for yellow? Could go either way, and then you're recovered. And then we applied, this is theoretical criteria from this paper, but then we actually applied standardized criteria with the scales and the various measures we had in patients at 6 months. So according to our criteria, everybody started, they were inpatient, they were in this sort of partial relapse state because they were weight restored, but so they're partly well, but they're not fully well for hospital discharge. And then we followed them at 6 months. And what you'll notice is that 29%, 30% were relapsed at 6 months, which matches 50% at 1 year, right? The question is, can we use any of the data from this, the modeling data, the perceptual data, to predict who these people are going to be so that we can better focus on them, give them maybe more intensive treatment, keep them inpatient longer, try novel treatments, et cetera. And what we found was that we were able to make some predictions in terms of who is more likely to relapse based on signals including the initial prior, the response bias, as well as the degree to which they rated stomach unpleasantness during the stimulation. So the more, the less they felt, the more aversive the stomach signals were, the more likely they were to relapse. This scale, this is sort of using a relapse criteria. We also just wanted to use dimensional sort of rating scales. If you just fill it out, you don't have to do anything fancy. We also found that this is the EDEQ global score over here. Lower scores are associated with better symptoms. So you can see like these people went into partial recovery. And we were able to also predict questionnaire scores using these various measures over here. We didn't see any differences in the brain, even though we found that both the patients and healthy comparisons showed neural responses. We did see kind of a larger envelope. So in this same posterior medial region of the brain, we saw that the anorexia nervosa patients actually had a much larger window. This is about 340 milliseconds. So their brain was sort of responding for a longer period of time than the healthy comparisons. So this might be something to follow up on, although I think from a clinical standpoint, the behavioral markers are probably going to be more useful at this point. Okay, so back to summary and take-home points. Hopefully, do you feel like you know a little bit more about interoception now? Yeah. Okay, that's good. Even if you just name recognition for the concept. Hopefully, I've shown you all of these sort of statements, particularly that controlled perturbations of interoceptive signals are an optimal way to reveal how these internal sensations are encoded across the body. And if you're interested, or you have a family member who says, "Hey, what is this term?" There's a book that just came out. This is actually, image was taken from the cover of it. That's actually written with a general population audience in mind, general readership. So again, I'd like to thank everybody, and especially thank you for listening. I'll take questions.
Any questions?
Thank you very much for this talk. So, one question I have, and again, because I work here mostly on like an in vitro model, and is, given the complexity of your research, is like how could some of those things could be done in vitro rather than doing it in patients, and even if it's relevant to do so, but also the other thing is what would be some sort of the outcome measures that you would do in vitro, and I think I want to relate that to what you show with our patient reported outcomes, which also how does that play a role in how you analyze or interpret the result, because we know how inter-patient variability can happen, and again, how can this relate to in vitro research to help you guys out in that sense?
It's a great question. So my work is, even though most of everything essentially from my lab that I've shown you is human, my work is starting to delve more into animal models, mainly through collaborators. At UCLA, I go to lab meeting with my colleagues in cardiology who do animal work, and it greatly informs my thinking. So going down to an in vitro, of course, it depends on the techniques that you use. But I know that organoids are certainly something that's an active area here. So think of the simplest interoceptive neural circuit could be, a peripheral organ cell and a neural cell, right? All of that complexity of hierarchical circuitry, you can study something in terms of maybe, two cells or two cells with an interneuron, so to speak, to see at a very simple level how some of the trafficking might develop. How does that differ from different cells from different internal organ systems? Animal models, mouse models, there's such great sophistication and interest from really stellar groups, including folks like Diego Bojorquez and Maya Kalber, right? Zach Knight, like very precise studies looking at gut sensing of nutrients, looking at brain sensing of thirst and osmolality changes. I think you want to be able to break it down to the simplest circuit. You want to be able to look at animal models that are more, I don't know if you would consider that to be in vivo, but certainly ones where you can have more precise perturbations, manipulations of a particular circuit. And then there are people in the primate world who are doing some of this work. You can sort of pick your evolutionary question. My lab's mascot is actually an octopus. I think there's some cool studies that you could do in octopus that, humans and octopi share a evolutionary ancestor 550 million years ago, but clearly they show evidence of embodied intelligence and the ability to sort of have a nuanced sense of what's going on in the body and to use it in a very sophisticated way. So I think it would come down to, right, what's the next question and what's the best sort of tool to address that question. But I actually see a lot of options. I even see in silico options. So we live now in the age of AI, and there's lots of, AI doesn't have a body, but you interact with an AI agent, an LLM, and there's certainly eventually a sense that there's going to be some degree of sentience, right? Super intelligence, whatever it is. How does pairing that with some degree of body sensing, so to speak, whatever the body of an LLM would be? I think there's a lot of open questions out there, and if you're interested in it, I'd be happy to talk, because I love thinking about this, and particularly with the nuanced sophistication at the level that you're talking about. I think there's many, many fruitful open questions. Yeah.
Great talk. My question is more related to the application of your findings into your clinical practice.
Yeah.
In what type in your psychiatric interaction with your patient, and the questions you ask and the responses you get, what things do you take and change your strategy towards the patient based on what you studied?
Sure. Can I spend 2 minutes showing you something different that wasn't in the talk? So, and this is directly in terms of the question about translation, right? So, one answer would be, in an ideal world, there would be an ingestible device that I can use for an eating disorder patient, right? That I can both maybe make a prediction about illness outcome, or I can use therapeutically to train them how to more accurately sense bodily signals. Of course, there are many, many different types of ingestibles out there. That doesn't have any broadcasting. It doesn't have any there's a whole another conversation in terms of what's actually possible there. But that doesn't actually translate right now into my clinic. I'll show you something that actually has translated into my clinic. And it's taking a completely different approach. Instead of stimulating interoceptive signals, or sensory signals, it's actually taking things away. So this is a float tank, flotation rest. It's called reduced environmental stimulation therapy. Right across, so the Terasaki facility in Westwood, right next to the corner bakery, there used to be a float facility. It's now closed. It was called Float Lab. Theirs didn't look like this, but this is a environment that is highly engineered. It's a room that is soundproof. It's lightproof. So when you lay down, it's 11 inches of water. When you lay down in the water, it's called floating because there's 2,000 pounds of Epsom salt, bath salt, more salt than the Dead Sea, so your skin doesn't prune, but you float effortlessly on the surface. You don't have to hold your breath. You don't have to tense a muscle. And you turn the lights off, so you basically attenuate visual and auditory input. It's the water and the air are heated, so it's the same temperature as the surface of your skin. So almost like an external hypothalamus in a way. So if you're attenuating visual input, you're attenuating auditory, you don't have to move a muscle, so there's no proprioceptive input. You're not touching the walls because you're self-centered. You're not moving. You're not speaking to anybody, right? What's left? We thought there'd be interoceptive signaling, and we've shown that in a variety of studies that your heartbeat and your breathing sensations are increased, and your gut sensations don't change. It has an anxiolytic, a strong anxiolytic effect. So this is an RCT, phase 3 style RCT we did. Cohen's D of 1.5. That's the same effect you get with a healthy dose of a benzodiazepine like Ativan or Xanax or Klonopin, like a strong dose, with no drug whatsoever, right? It's a reliable effect, and what we found is that we were actually able to see a small but reliable change in body image perception, okay? So body image, which is one of the core features. So, this is the evidence showing that it increases your breath and heartbeat sensations, but doesn't change or even decreases the stomach, and also increases the valence. So people actually feel their heartbeat, their breath, and their stomach more positively when they're in this environment. So the one thing that I've actually been able to do clinically as a psychiatrist currently, I have patients who I don't recommend that everybody float, but when it seems relevant, particularly when people are in the early phase where anxiety meds like benzodiazepines are not indicated or they haven't responded to those, I suggest that they try floating, and we've seen some positive efficacy there. We're studying it. My former postdoc has a K award where we're combining it with psychotherapy to try to kind of augment that. And it's early days, but there's definitely some applications there. From a biomarker standpoint, what does this do to the brain? What does this do to peripheral signals? What does this do to EEG? What does this do to cortisol? What, serotonin? All that. Interesting questions that could be asked there too. Okay. Thank you for the extra time.
One quick question. A lot of mental disorders are related to trauma.
Yeah.
Are there efforts to unwire the brain to erase that memory or to cure?
Yeah, so I've written grants to try to use floating as a way to help people cope with the traumatic memory associated with. Some people will actually spontaneously have recovery of those memories. The goal in trauma-related therapy isn't to try to erase that memory. It's not possible, right? Any more than you can, you just can't erase these memories, except for maybe, if you have developed Alzheimer's dementia in late stage, right? The goal is to better understand what are the processes that reinforce that trauma. And one of the theories that we wrote a paper on this is that, traumatic experiences, they're events. It's learning that happens. Something happens to you in a distinct period of time that stamps you. And people then have traumatic recall. So if you're in a war zone, head injury or smells, they can teleport, loud sounds, smells, they can teleport you back. And because that was such a high stress, high fear state, people can reactivate fear axis, HPA axis signaling in their body. And we have a theory that part of what happens to maintain the negative aspect of that memory is that as the fear memory reactivates the body, they also learn to become afraid of the body's sensations themselves. So they actually begin to avoid anything that increases that heartbeat activation, for example. And one of the reasons why I'm interested in studying float therapy for trauma is that because, yes, you do feel those sensations more strongly, but it's more pleasant. And so can you help people, rather than trying to erase, say, yeah, here's the memory, but here's now a new memory of a more positive experience of the body. And of course, there's a lot of other psychological and medication stuff that you need to do. But if you can facilitate that new learning process, you can then balance the negative effects of the trauma learning or the trauma memory.
Are there biomarkers for flight versus fear-flight syndrome that you can counterbalance that body has?
There are people who've looked at this, and in terms of CRH antagonists in the brain and PACAP signaling, et cetera. Big trials, none of them have panned out so far. Even though there's a decent neurobiology that seems to be female selective, somehow it's more specific. The trials so far haven't yielded a result. Thank you. It's a great question.
Any other question?
Great, thanks. Yeah. A couple questions. First is a more strategic question. I was just impressed by from the anorexia study you did, right, and then adding more patients. The backdrop. I was at J.P. Morgan meeting, and the reason I was meeting the ARPA-H program officers.
Sorry,
I was meeting with the ARPA-H program officers,
yeah.
and from what they have new program going to come out, for example, on new wearables and things. But now with the new director, there's a much more emphasis on getting to human fast, right? So if I look at the kind of the patient population you have, actually providing a potentially studies that you can get device to patient much faster. With that said, I'm just kind of curious, is it difficult to recruit patients, or in this kind of patient population, it's much easier to recruit, versus the other disease indications?
So are they easy to recruit or difficult to recruit? This, in my experience, these are some of the best patient populations to work with for a few reasons. One is that from a personality standpoint, the way that these patients are very rule-abiding, just they're perfectionistic. They want to do, they want to do well. They want, when they participate in something, they're motivated. They're also understudied. So when there is somebody that's interested in understanding their pathology and helping it, I've actually found them to be sometimes easier to recruit than healthy comparisons. The caveat is it depends on the stage of illness that you're looking for. In this case, we're looking at weight-restored people. When they remain symptomatic, even when their body weight puts them out of a range where they would be typically considered anorexic from the AN diagnosis. They're still symptomatically severe. That study that I showed you at Liber, there was an inpatient eating disorder unit in the building, so the patients literally came down. So we were fortunate in that regard that we had a nice collaboration. But I've collected data from those patients in the LA area before, and there are many treatment centers. LA is a large metropolitan area, and I would not expect that it would be exceedingly challenging. We also have eating disorder programs at UCLA.
Great. And also the visions, we want to have a larger LA kind of collaboration, and that's kind of, that's one of the things actually when Paul Sheehan, the program officer, visit, he would like to see more LA kind of collaboration that put up these big proposals. That's good. The more technical questions, for Carl Destras, right, the optogenetic study seems, you identify that population of a specific zone that affect the interoception. Is there a way to target those? Right now, for those regions, is it the only way is pharmacological? Can you do things some like ultrasound? But it's always a challenge to go through the skull, like all the cranium stuff, probably not specific enough, right?
In other words, can you target those regions in human? Is that what you're saying? Yes. Yes. And in fact, I'm working with a collaborator where we're proposing to do just that. Now, you're right about so the question is, in reference to the findings from the Carl Destras group's optogenetic findings in the medial prefrontal cortex and the insula, can you target that in humans? So with focused ultrasound, there is an issue in terms of beam refraction, and the skull characteristics play a big role. Turns out the temporal bone is quite flat, and so it's actually a good window for doing things like trying to target the amygdala. It becomes a little bit more complicated when you try to target a region like the insular cortex, although it's possible. And I do think that that TMS, there is versions of TMS that people are actually using currently to try to get at the medial prefrontal piece. So I am optimistic in doing it. Obviously, we need the funding to do that. But the other piece that I think is really exciting potentially for focused ultrasound is that in his paper, they also had hypothalamic regions and brainstem regions. And what's unique with ultrasound is that you can actually get at those areas. There are people doing thalamic stimulations. To be honest, it scares me a little bit because anytime you start messing with core homeostatic control regions, I'm a little, but at the last conference, I was at the ACNP meeting last week, and there was somebody who was doing anterior thalamic stimulation. So, what happens if you hit the reticular nucleus of the thalamus and you shut down, a good chunk of the subnuclei? I don't know. But people are starting to do it, and that, for me, that helps to de-risk it. The question really is, what's the optimal target? And from a symptom perspective, in humans, it would seem to be most useful to start with regions like the insula and the medial prefrontal cortex, given the data that we have showing symptomatic associations. They're larger targets, but the other downside is that those cortical regions could be much larger than what you would target with focused ultrasound, whereas the brainstem and thalamic regions, you could actually conceivably try to hit, let's say, the NTS. I have no idea what happens in a human. I don't know if anybody's done an NTS stimulation study, because most of the ultrasound stimuli are inhibitory, it turns out. It seems like they have an inhibitory not neurotransmitter, but inhibitory effects. I don't know what happens in a human if you successfully shut down their NTS.
Great, great. And also a little anecdote. When I was at Stanford, actually, I know Carl Desra pretty well, and his postdoc, Al Borden and Feng Zhang, who the first one did optogenetics. And I told them, I said, what a stupid idea. How do you expect the fungal gene will work? But fortunately, they didn't listen to me. Otherwise, I'll be, but it's great. Another question is the, in your elegant Nature Neuroscience work, you showed, right, that sensation was going through different pathway, and there might be, some slow, some fast. Is there an understanding of how those signals gets integrated, right, like in the brain, in terms of, do you want to amplify one or the other, like how the brain decides to filter or weigh the different sensations, right? Like how there these studies try to understand the combination of the various interoceptive pathway for the same sensation.
For which signal are we talking about? The capsule stuff?
The one that, right, you have that patient that have what's the disease, right? So one
where we do the anesthetic, yeah.
Yeah. I'm just saying, like you're showing that
Right.
there's multiple pathways.
Right. So we don't have real good data for simultaneous pathway information. So it would probably be more speculation than anything else. What I skipped over was the idea that you do have this thing called a heartbeat evoked potential, which this is just a schematic circuit from a review that it's a nice review on this, where these are some of the areas where you actually in the brain where you actually see at a neuronal spiking level tracking of heartbeats. And people have done like electrocorticography, so putting grid electrodes directly on S1, putting depth electrodes directly in the insular cortex. Turns out they're everywhere. If you put it in VMPFC, OFC, amygdala, cingulate, you'll find subsets of neurons that respond to these heartbeat signals. And we actually published a paper using the same approach where we showed abnormal HEPs in generalized anxiety patients at that same 0.5 microgram dose. So this is showing that difference here within the groups. I think that there could be value to trying to maybe do science tracking that signal. Whether you could say, oh, this is an insular source or a somatosensory, I don't have any confidence in that. But given that there's so many disparate cortical regions that clearly sample, there's also regions that do respiratory sampling, and there's some neurons that do both. That was a paper from Olaf Blanke's group in Nature Communications 2 years ago. It means that, again, the brain prioritizes access, prompt, very rapid access to these signals, also glucose sensing and thirst sensing, in ways that I think allow it to make these interpretations and predictions. One of the things that I've been struck by is that thirst sensing, when you're completely dehydrated and you drink like a big glass of water, you stop drinking minutes before your blood osmolality recovers and you're no longer actually dehydrated. And how do you do that? There's a subfornical circuit, and there's sensors, orosensory, sensors in the mouth and the upper pharynx that project to SFO. This is Zach Knight's work, that literally tell you how much is gone, tell the brain how much has gone in, and now is a good time to stop. So it makes sense that there would be a lot of these rapid relays that are doing things almost automatically, not just for glucose and for thirst, but also for the heartbeat. And so maybe HEPs and EEG signals could be ways to listen in on that kind of more automated processing and see whether we can use that in a way that helps make clinical decisions, whatever those might be.
Oh, that's fascinating. You're saying these circuits are actually making projections, right? Not just passively sensing. And,
right.
And the last, just very quick question for anorexia patient, right? The most striking part is always how can that distort their visual perception, the visual perception. For the anorexic patient, how does that distort the visual perception, right, of their body. Like, how can someone see a skeleton of herself and think she's very,
Yeah.
very, overweight? Like, how does that circuitry could
Yeah.
work?
So my colleague, my former mentor and now colleague and collaborator, Jamie Fusner, who used to be at UCLA, studies visual perception in anorexia nervosa. And what he's shown is that, that you actually have evidence of abnormal visual stream, dorsal and ventral, abnormal activity in the visual stream in patients with anorexia nervosa when they look at visual information. And it's not simply the body. It's the ratio of low to high frequency stimulation. And so one of the theories, and we actually wrote a paper last year on the sort of relative role of the insula and visual input related to body image. It's a theory, but it's based on some of his data, where essentially if you are abnormal in your ability to sense visual input, and you're abnormal in your ability to sense interoceptive input, right? You have a deficit in one area and you have more of a focus in the other because you have difficulty perceiving it. Maybe people have become more fixated on visual input and less fixated on interoceptive input, and that affects visual and insula specific circuits. And if you can shift that imbalance, using floating was maybe one example. He's currently studying perceptual retraining, where you actually train people to discriminate the signal that they have difficulty looking at. You might be able to then begin to reshape that circuit and reduce them.
But when they see other people, they see fine, right?
Exactly.
Only themselves. So
Exactly.
some switch turned on, like
Correct.
some neuron firing to distort the vision.
Correct. Yeah, when they look at other, like if they look at somebody else who is very underweight, they would say accurately, oh, they're very underweight. But when they're looking at an image of themselves that might have the same low body weight, they would see, and report anyways, they would perceive a different image, even though the visual input is the same. And again, the link there is that it's self-related, and we think that it has something to do with insula and interoceptive circuitry that is abnormal. Like when it relates to self-perception, it's distorted. Yeah, great question.